Golgi apparatus, right? Um, the minute details about this organelles we have studied, right? Today we'll be studying about mitochondria, vacuole, plastids. Okay, so we'll start with mitochondria, the energy currency of the cell, right? The powerhouse of the cell. Basically, it's a double membranous structure. Mitochondria, slightly you can say disc or oval kind of shape. Outer membrane is little porous, inner is little rigid and modified into many infolds. Every single infolding of the inner membrane is called crista singular, cristae plural. Okay, so about mitochondria, no doubt all the cellular activities, all the chemical cellular activities which take place inside the complete cell. For that, the energy is supplied by the mitochondria. So mitochondria definitely inside it also should have various kinds of enzymes, various kinds of other substances which help in the generation of energy rich molecules called as ATP or adenosine triphosphate. Okay. So this all the what you can say energy needed for the life of the cell is supplied by mitochondria, but for that mitochondria itself should be self-sufficient, and that is the reason a very strong, strange, unique feature of our mitochondria is it has its own DNA. It has its own ribosomes. This is very nice thing about mitochondria. That is, when it has its own DNA, when it has its ribosomes, that means definitely it can manufacture some of the types of proteins, right? Then it again undergoes cell division. It can go with its own division by fission. Okay? By fission method, it divides by fission. Again, if you look at the structure, the outer structure, as it is a membranous structure, and again the number of mitochondria available in a particular cell will be variable depending upon again the kind of cell, the kind of activity in which the cell is involved. Okay, outer membrane porous compared to the inner membrane. Inner membrane turns into infolds. These infolds are called cristae. These two membranes divide the internal structure of mitochondria into two compartments, right? The inner compartment is filled with the fluid of mitochondria that's called the matrix, rich in various substances, glucose, sugars, glycoproteins, and enzymes. Okay, and most important, you can say that the folds here of the inner membrane of mitochondria, the folds actually they provide large surface area for the chemical activities in the formation of ATP. More folds inside, that means more surface area is made available. Okay, these folds are called as cristae, and I said it's singular crista. So mitochondria definitely an important cell organelle, because this is the only organelle which provides energy to the cell, which carries out the cellular respiration, right? So singular mitochondrion, plural mitochondria. Then again, one important cell organelle, little bit in structure just like mitochondria, is the plastids. Plastids, you all know, they are present only in the plant cell first thing. In general, you can divide them into two categories. In higher grade students, you will be studying with one more form of plastid is also there, that is chromoplast. In general, two, that is chloroplast and leucoplast. You all know that chloroplast is the place wherein the chlorophyll pigment is present inside that flat disc-shaped sacs called as thylakoids, right? So chloroplast is one kind of plastid wherein chlorophyll is present. That means there actually the process of photosynthesis takes place, right? Because of the chlorophyll pigment. There are other kinds of chloroplasts also here available, that is the xanthophyll, carotene. Those pigments are also there. Then another kind of plastid completely responsible for the storage and not having any color, colorless or then say creamish kind of color. That kind of plastid is called leucoplast. It also can be divided into three more division on the kind of storage of food, kind of material it stores. So leucoplast students basically involved in the function of storage. Chloroplast Chloroplast basically for photosynthesis, right? Leucoplast storage of food. Best example, potatoes you can take. Rich source of starch content, right? Sweet potatoes. So they are storage organs. The kind of pigment plastic present is the leucoplast, responsible for the storage of starch then, right? So leucoplast completely responsible for the storage. It is not alone storing the starch, it also stores proteins, it also stores oils or fatty acids, right? So leucoplast completely a kind of plastic responsible for storage. Chloroplast on the other hand, you know that chloroplast is the place wherein the process of photosynthesis takes place because the pigment chlorophyll is present there. Plastic is present only in plant cells, right? Again, like I said, it is somewhat similar to a mitochondria. That means it has DNA and ribosomes. Plastids have their own DNA, they have their own ribosomes. Means to a certain extent, they can carry out protein synthesis because of the presence of ribosomes. The next important term is about vacuoles. You have studied that. Vacuole is what? A bag, a sac, which can store anything in the liquid form, in the solid form. It is primarily present in the plant cell, bigger in size. In animal cell, negligible or very small size vacuoles are seen. Plant cell basically they make use of the vacuoles or the sac as a storage department, storage organ, storage unit. Storage of it can be amino acids, it can be sugar, it can be proteins, it can be anything else other than that. Useful substances. Okay, in case of amoeba, a big vacuole, you know that in the structure you have seen in the diagram. That big vacuole stores the food it has consumed, right? Basically, vacuoles are also responsible for eliminating or expelling excess water from the cell or even waste of the cell, which is not required by the cell, even that is eliminated through the vacuoles. And vacuoles, you know, in case of some plant cells, they form the major part of the cell. In fact, 50 to 90% of the volume of the cell is occupied by a vacuole. That means if I draw a plant cell here, so in that case, plant cell means I have to draw a cell wall, right? Right? Somewhere here, if the nucleus is there, I won't draw the other cell components, but the vacuole somewhat will be such big in size. So in many plant cell, vacuole occupies more than 50% of the cell volume. In some cases, 90% of the cell volume. Again, depends to what extent it is acting as a storage unit, right? So vacuoles more prominent, bigger size, all in plant cells. Then storage of what you can say amino acids, sugars, and all that we discussed. Most important, they provide rigidity, turgidity to the complete cell. Again, the wall of the vacuole, the membrane of the vacuole is called tonoplast. Tonoplast provides rigidity to the surface of the vacuole. It does not easily get burst inside the cell because of the outer membrane, that is tonoplast. We will be studying about tonoplast and all in higher grades. Okay. So students, these are some of the important cell organelles. Some of them are seen prominently in the plant cell, while some in the animal cell. Apart from this, there is centrioles and centromere, responsible for the cell division, which is found only in the animal cell, not in the plant cell. Then in case of nucleus, we have studied nucleus has nuclear pores, the genetic material in the form of what you can say chromatin. When it gets organized properly in any of the cell division state, then it is called chromosomes. So that means basically genetic material is present in the nucleus. The to and fro from the nucleus to the surrounding cytoplasm, from there inside the nucleus, it takes place through some way should be there from the nucleus, that is through the nuclear pores, right? So through nuclear pores, the incoming, outgoing work goes on from the nucleus to the cytoplasm, from the cytoplasm to the nucleus. This all goes on. So about every single cell organelle we have discussed in detail, right, students? So what you people are supposed to do is you are going to make a note of the structure, function along with the diagram of endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, plastics, and vacuoles. So this is your homework. Okay, you can take some almost 3 days of time for completion. Until Monday, you can submit it to me. Fine. So in my next session, we'll be studying about another interesting topic. Fine, students.